So, hoping you have understood the last three parts, let us talk about cytoplasm of the cell. The cytoplasm is filled with minute and large dispersed particles and organelles. The jelly-like fluid portion of the cytoplasm in which the particles are dispersed is called cytosol and contains mainly dissolved proteins, electrolytes, and glucose. Dispersed in the cytoplasm are neutral fat globules, glycogen granules, ribosomes, secretory vesicles, and 5 especially important organelles: the endoplasmic reticulum, the Golgi apparatus, mitochondria, lysosomes, and peroxisomes. All these organelles are very important and have separate functions attached to them. Starting off with endoplasmic reticulum, which is a network of tubular structures called cisternae and flat vesicular structures in the cytoplasm. This organelle helps process molecules made by the cell and transports them to their specific destinations inside or outside the cell. The tubules and vesicles interconnect. Also, their walls are constructed of lipid bilayer membranes that contain large amounts of proteins, like the cell membrane. The total surface area of this structure in some cells, the liver cells, for example, can be as much as 30 to 40 times the cell membrane area. The space inside the tubules and vesicles is filled with endoplasmic matrix, a watery medium that is different from fluid in the cytosol outside the endoplasmic reticulum. Electron micrographs show that the space inside the endoplasmic reticulum relates to the space between the two membrane surfaces of the nuclear membrane. Substances formed in some parts of the cell enter the space of the endoplasmic reticulum and are then directed to other parts of the cell. Also, the vast surface area of this reticulum and the multiple enzyme systems attached to its membranes provide the mechanisms for a major share of the cell's metabolic functions. What is ribosomes and the rough, or granular, endoplasmic reticulum? Attached to the outer surfaces of many parts of the endoplasmic reticulum are large numbers of minute granular particles called ribosomes. Where these particles are present, the reticulum is called the rough, granular, endoplasmic reticulum. The ribosomes are composed of a mixture of RNA and proteins. They function to synthesize new protein molecules in the cell. Then what is smooth, or agranular, endoplasmic reticulum? It is part of the endoplasmic reticulum, has no attached ribosomes. The smooth reticulum functions for the synthesis of lipid substances and for other processes of the cells promoted by intra-reticular enzymes. Next up is Golgi apparatus. The Golgi apparatus is closely related to the endoplasmic reticulum. It has membranes like those of the smooth endoplasmic reticulum. The Golgi apparatus is usually composed of 4 or more stacked layers of thin, flat, enclosed vesicles lying near one side of the nucleus. This apparatus is prominent in secretory cells, where it is located on the side of the cell from which secretory substances are extruded. The Golgi apparatus functions in association with the endoplasmic reticulum. Small transport vesicles, also called endoplasmic reticulum vesicles or ER vesicles, continually pinch off from the endoplasmic reticulum and shortly thereafter fuse with the Golgi apparatus. In this way, substances entrapped in ER vesicles are transported from the endoplasmic reticulum to the Golgi apparatus. The transported substances are then processed in the Golgi apparatus to form lysosomes, secretory vesicles, and other cytoplasmic components. Let's talk about lysosomes. Lysosomes are vesicular organelles that form by breaking off from the Golgi apparatus. They then disperse throughout the cytoplasm. The lysosomes provide an intracellular digestive system that allows the cell to digest the following: damaged cellular structures, food particles that have been ingested by the cell, and unwanted matter such as bacteria. Lysosomes are different in various cell types, but are usually 250 to 750 nanometers in diameter. They are surrounded by typical lipid bilayer membranes and are filled with large numbers of small granules, 5 to 8 nanometers in diameter, which are protein aggregates of as many as 40 different hydrolase digestive enzymes. A hydrolytic enzyme can split an organic compound into two or more parts by combining hydrogen from a water molecule with one part of the compound and combining the hydroxyl portion of the water molecule with the other part of the compound. For example, protein is hydrolyzed to form amino acids, glycogen is hydrolyzed to form glucose, and lipids are hydrolyzed to form fatty acids and glycerol. Hydrolytic enzymes are highly concentrated in lysosomes. Ordinarily, the membrane surrounding the lysosome prevents the enclosed hydrolytic enzymes from meeting other substances in the cell, and therefore prevents their digestive actions. However, some conditions of the cell break the membranes of lysosomes, allowing release of the digestive enzymes. These enzymes then split the organic substances with which they come in contact into small, highly diffusible substances such as amino acids and glucose. Let us also discuss peroxisomes. Peroxisomes are physically similar to lysosomes, but they are different in two important ways. First, they are believed to be formed by self-replication, or perhaps by budding off from the smooth endoplasmic reticulum, rather than from the Golgi apparatus. Second, they contain oxidases rather than hydrolases. Several of the oxidases are capable of combining oxygen with hydrogen ions derived from different intracellular chemicals to form hydrogen peroxide. Hydrogen peroxide is a highly oxidizing substance and is used in association with catalase, another oxidase enzyme present in large quantities in peroxisomes, to oxidize many substances that might otherwise be poisonous to the cell. Secretory vesicles is one of the important functions of many cells is secretion of special chemical substances. Almost all such secretory substances are formed by the endoplasmic reticulum, Golgi apparatus system, and are then released from the Golgi apparatus into the cytoplasm in the form of storage vesicles called secretory vesicles or secretory granules. These vesicles store protein proenzymes, enzymes that are not yet activated. The proenzymes are secreted later through the outer cell membrane into the pancreatic duct and then into the duodenum, where they become activated and perform digestive functions on the food in the intestinal tract. Now the next organelle, which is mitochondria. The mitochondria are called the powerhouses of the cell. Without them, cells would be unable to extract enough energy from the nutrients, and essentially all cellular functions would cease. Mitochondria are present in all areas of each cell's cytoplasm, but the total number per cell varies from less than 100 up to several thousand, depending on the energy requirements of the cell. Cardiac muscle cells, cardiomyocytes, for example, use large amounts of energy and have far more mitochondria than fat cells, adipocytes, which are much less active and use less energy. Furthermore, the mitochondria are concentrated in those portions of the cell responsible for the major share of its energy metabolism. They are also variable in size and shape. Some mitochondria are only a few hundred nanometers in diameter and are globular in shape, whereas others are elongated and are as large as 1 micrometer in diameter and 7 micrometers long. Still others are branching and filamentous. The basic structure of the mitochondrion is composed mainly of two lipid bilayer protein membranes, an outer membrane and an inner membrane. Many enfoldings of the inner membrane form shelves or tubules called cristae, onto which oxidative enzymes are attached. The cristae provide a large surface area for chemical reactions to occur. In addition, the inner cavity of the mitochondrion is filled with a matrix that contains large quantities of dissolved enzymes necessary for extracting energy from nutrients. These enzymes operate in association with oxidative enzymes on the cristae to cause oxidation of nutrients, thereby forming carbon dioxide and water, and at the same time releasing energy. The liberated energy is used to synthesize a high-energy substance called adenosine triphosphate, ATP. ATP is then transported out of the mitochondrion and diffuses throughout the cell to release its own energy wherever it is needed for performing cellular functions. Mitochondria are self-replicative, which means that one mitochondrion can form a second one, a third one, and so on whenever the cell needs increased amounts of ATP. Indeed, the mitochondria contain DNA, similar to that found in the cell nucleus. The DNA of the mitochondrion plays a similar role, controlling replication of the mitochondrion. Cells that are faced with increased energy demands, for example, in skeletal muscles subjected to chronic exercise training, may increase the density of mitochondria to supply the additional energy required. Now let's talk about cell cytoskeleton, which is filament and tubular structures. The cell cytoskeleton is a network of fibrillar proteins organized into filaments or tubules. These originate as precursor proteins synthesized by ribosomes in the cytoplasm. The precursor molecules then polymerize to form filaments. As an example, large numbers of actin microfilaments frequently occur in the outer zone of the cytoplasm, called the ectoplasm, to form an elastic support for the cell membrane. Also, in muscle cells, actin and myosin filaments are organized into a special contractile machine that is the basis for muscle contraction. Intermediate filaments are generally strong rope-like filaments that often work together with microtubules, providing strength and support for the fragile tubulin structures. They are called intermediate because their average diameter is between that of narrower actin microfilaments and wider myosin filaments found in muscle cells. Their functions are mainly mechanical, and they are less dynamic than actin microfilaments or microtubules. All cells have intermediate filaments, although the protein subunits of these structures vary, depending on the cell type. Specific intermediate filaments found in various cells include desmin filaments in muscle cells, neurofilaments in neurons, and keratins in epithelial cells. A special type of stiff filament composed of polymerized tubulin molecules is used in all cells to construct strong tubular structures, the microtubules. Another example of microtubules is the tubular skeletal structure in the center of each cilium that radiates upward from the cell's cytoplasm to the tip of the cilium. Also, both the centrioles and mitotic spindles of cells undergoing mitosis are composed of stiff microtubules. A major function of microtubules is to act as a cytoskeleton, providing rigid physical structures for certain parts of cells. The cell cytoskeleton not only determines cell shape, but also participates in cell division, allows cells to move, and provides a track-like system that directs the movement of organelles in the cells. Microtubules serve as the conveyor belts for the intracellular transport of vesicles, granules, and organelles such as mitochondria. Nucleus. The nucleus is the control center of the cell and sends messages to the cell to grow and mature, replicate, or die. Briefly, the nucleus contains large quantities of DNA, which comprise the genes. The genes determine the characteristics of the cell's proteins, including the structural proteins, as well as the intracellular enzymes that control cytoplasmic and nuclear activities. The genes also control and promote cell reproduction. The genes first reproduce to create two identical sets of genes. Then the cell splits by a special process called mitosis to form two daughter cells, each of which receives one of the two sets of DNA, genes. Unfortunately, the appearance of the nucleus under the microscope does not provide many clues to the mechanisms whereby the nucleus performs its control activities. During mitosis, the chromatin material organizes in the form of highly structured chromosomes, which can then be easily identified using the light microscope. Nuclear membrane. The nuclear membrane, also called the nuclear envelope, is actually two separate bilayer membranes, one inside the other. The outer membrane is continuous with the endoplasmic reticulum of the cell cytoplasm, and the space between the two nuclear membranes is also continuous with the space inside the endoplasmic reticulum. The nuclear membrane is penetrated by several thousand nuclear pores. Large complexes of proteins are attached at the edges of the pores so that the central area of each pore is only about 9 nanometers in diameter. Even this size is large enough to allow molecules up to a molecular weight of 44,000 to pass through with reasonable ease. Nucleoli and formation of ribosomes. The nuclei of most cells contain one or more highly staining structures called nucleoli. The nucleolus, unlike most other organelles discussed here, does not have a limiting membrane. Instead, it is simply an accumulation of large amounts of RNA and proteins of the types found in ribosomes. The nucleolus enlarges considerably when the cell is actively synthesizing proteins. Formation of the nucleoli and of the ribosomes in the cytoplasm outside the nucleus begins in the nucleus. First, specific DNA. Genes in the chromosomes cause RNA to be synthesized. Some of this synthesized RNA is stored in the nucleoli, but most of it is transported outward through the nuclear pores into the cytoplasm. Here it is used in conjunction with specific proteins to assemble mature ribosomes that play an essential role in forming cytoplasmic proteins. That was all about cell and its structure. Stay tuned for more.
Have you ever heard of video books? sqadia.com is delighted to present video books, medical books that have been tailored into a series of videos. sqadia.com videos are presented in the same order as the book's chapters. Explore now and make your learning easy.